shall now proceed and look at the phototransduction. This is supposed to be phototransduction. Okay, so under phototransduction, we are looking at how these light will be converted into the electrical signals that will be sent to the brain. So electrical signals which will be sent to the brain. So what happens here is that we shall have bleaching of rhodopsin, and briefly talking about the rhodopsin. So rhodopsin, we know that it's the photopigment found in the rods. Rhodopsin, and it has one form. It's not like the opsins which are found in the cones that exist in three forms. So the rhodopsin, we know that its wave length, it absorbs light in the wavelength of approximately that 505, whereas you find that for cones they will have in the range of like 440 something, then you have 535, and you have 570. So you have 445, you have 535, then 570. We know that red is for the highest, then we have green, and then we have blue having the least wavelength. So that's it for cones. But rhodopsin has a fixed wavelength which it absorbs, that's 505. And then the other thing we need to know is that what makes up this rhodopsin. Rhodopsin is made up of two components. We have the opsin itself, that is scotopsin. Scotopsin, I can correct that. Scotopsin is the protein. Then it has the other one. The other component is the retinol. But this retinol is in the form of 11-cis 11-cis form. For those who did chemistry, we can tell the difference between cis and the trans. So it exists as the 11-cis retinol. That's if it's combined with the scotopsin. That's the form that combines with the scotopsin to form rhodopsin. So what happens when light strikes this rhodopsin? Rhodopsin, whenever it's struck by light, it will undergo changes, which changes will in turn affect the ion channels in this rod cell. Okay? So we have rhodopsin here, which is the combined scotopsin and the 11-cis retinol. So when it's struck by light, it will change to a form. Let's have a short form here, which we can refer to. The short form is BLM. So when it's struck by light, it will change to B. That is batho-rhodopsin. You get batho-rhodopsin. You can add rhodopsin. Then from batho-rhodopsin, it takes a few milliseconds or microseconds, which details you may not need, to change to another form, which is litho-rhodopsin. That's a litho-rhodopsin. Then from litho-rhodopsin, it changes to meta-rhodopsin. All this occurs at a very fast rate that you may not recognize the changes. So this is meta-rhodopsin, but meta-rhodopsin has two forms. We have meta-rhodopsin 1 and meta-rhodopsin 2. So it first changes to meta-rhodopsin 1, and eventually it changes to meta-rhodopsin 2. And this meta-rhodopsin 2 is what we actually call the activated rhodopsin. You get the point? So this, when it's struck by light, it goes through the changes to become activated. So activated rhodopsin is known as meta-rhodopsin type 2. You get? So that's meta-rhodopsin type 2, which is known as the activated rhodopsin. So this meta-rhodopsin type 2 is actually the one which will cause the changes that will occur and in turn affect the ion channels within this. So what happens is that this meta-rhodopsin will spontaneously break down into two the components of the rhodopsin, that is the scotopsin will be formed, scotopsin, and then the other one, the dissociated rhodopsin doesn't remain as 11-cis, but in other words, it changes to all trans retinol. You get? All trans retinol. That's in the dissociated form. That's how it will turn out to be. So this scotopsin now will be the one that will drive further reactions that will occur. So what will it do? If we look at our rod cell, it has different ion channels. We have what I've called here the cyclic GMP gated sodium ion channels, meaning they have cyclic GMP on them, but they are primarily for allowing movement of sodium ions. Where? Into the rods. You get? The rod cells. So it's for moving sodium ions into the rod cells. Whereas this side, this is the outer segment where I have these cyclic GMP gated sodium ion channels. That is in the outer segment. Whereas in the inner segment, we have this sodium potassium ATPase pump. That's in the inner segment. But we not only have that pump, we also have these potassium ion channels that are selectively permeable to potassium ions, and they are not gated. So those are potassium selectively permeable channels, potassium ion channels, all both in the inner segment. Then we saw this is just the cell body where I have the nucleus and then the synaptic part of it. So back to the point, we are saying that we have scotopsin, which is going to cause the changes. So we must know what happens before we have the light. Initially, if we don't have light, let's assume we are in darkness, we have this potassium ATPase pump working, and we are pumping out sodium ions, as you can see the arrow facing outward, then potassium ions inward. Some of these potassium ions that are pumped inward will move out through the potassium and the selectively permeable potassium ion channels. You get? And then in the outer segment, we shall have sodium ions moving in to compensate for what was pumped out this side. You get? That is in dark. However, when we have light coming in, it will reverse everything. So the effect of light is that you will have scotopsin activating a G protein. This G protein is known as transducin. You get? So the G protein is known as transducin. That's a G protein. And that transducin will in turn activate another enzyme, which is known as PDE, specifically cyclic GMP PDE. So what is CGMP PDE in full? It is cyclic guanosine monophosphate phosphodiesterase. This one will just work to break down these cyclic GMP channels so that you don't have any sodium ion moving inward. You get? So when you have the phosphodiesterase being activated, it means that you cut off any entry of sodium ions in the outer segment. However, you will not have affected the inner segment where we have the potassium base pump still working. So what will this cause? It will cause a decrease in the transfer of sodium ions in the outer segment. However, we still have pumping of sodium ions in the inner segment outside of the cell. Get? So what will be the effect of this? The effect of this will be that you will have hyperpolarization because you are reducing the total or the net positive charge within the cell. So you will cause hyperpolarization. But you can't talk about hyperpolarization before knowing the normal potential of the cell. So the normal potential of the cell is initially 40. That is, I'm assuming we are in the dark and we don't have any interruptions. It's initially around negative 40 millivolts. But when you bring in the light and causing all these changes, it can reduce to about negative 70 or negative 50 millivolts. So this is what we refer to as the hyperpolarization. So the hyperpolarization will in turn have its own effects in the transfer of the signals. So again, we must know what happens in the dark before we have the light. Without the light, we have initially release of the inhibitory signals, which are specifically a neurotransmitter known as glutamate. So in the dark, you have glutamate being released, and it's inhibitory glutamate. So this is in the dark. However, when we have hyperpolarization coming in, you will reduce the amount of glutamate being released, which means that now you will have transfer of the sensory signals through the bipolar cells. Later on, we are going to look at the types of bipolar cells and the following, like ganglion cells, their types. So we have seen that initially in the dark we have depolarization and there is release of glutamate, which inhibits transfer of these signals. Then when we bring in light, it causes hyperpolarization, reduce the formation of or release of glutamate from these photoreceptor cells, and that would call for transfer of the sensory signals. So we can now briefly talk about how this transfer will occur. So the transfer of these visual signals doesn't occur like any other sensory organ, but we have what we call electrotonic conductions. Electrotonic conductions. So what are electronic conductions? For those of us who did biology, we know very well that we have what we call all or nothing rule. So here it does not apply to the photoreceptors. They will just transfer these visual signals, in that the higher the light intensity, the higher the amount that will be transferred, whereas the lower the light intensity, that will be the lower effect. You get? That's what we refer to as electrotonic conductions. So that's how these signals will be transferred as electrotonic conductions. However, when we reach the ganglion cells, in the way we looked at the structure of the retina, when we reach the portion of ganglion cells, they differ from these other cells in that for them, they will now generate the action potentials. So take note that action potentials are only generated at the level of the ganglion cells, whereas the other cells, the visual signals are transferred as electrotonic conductions. So that will cover the bit of phototransduction. And in the next part of this video, we are going to look at the different types of cells, and that is the bipolar cells and the ganglion cells within